Hello, good evening to the participants. On behalf of Growing Seed, I would like to welcome you all to the eighth session of the uh, workshop on biological data analysis and bioinformatics. So, past days we have learned about biological data analysis and bit in bioinformatics also. So, today the session will be taken by Dr. Sanghamitra Rout, Assistant Professor, and she will take classes on data analysis, special focus on PCA. So, Dr. Raut, I would like to transfer the call to you. Kindly start the session. Thank you all.
Am I audible now?
Yeah, you are audible.
Yeah. So, good evening, everyone. My name is Dr. Sanghamitra Rout. I'm working as assistant professor in CUTM University, Odisha. So, today I'm going to cover up two topics, that is principal component analysis and cluster analysis. So, these two things are very important for any statistical analysis to be done. So, I'm going to share the screen. My screen is visible. Yes, from the audience, can anyone say?
Yeah, it's visible.
Yeah, fine. Thank you. So, in this workshop, actually, I want to tell about this principal component analysis and why it is being done, first and foremost thing. Then, what is the utility of it? What could be the application? When we are having a huge data set, whether we can go for this principal component analysis or not. Then, in R, how can we do this analysis? So all these things I'm going to cover up in my presentation. Along with this theoretical presentation, I will run the codes in the R software for your easy way to calculate the principal component analysis of your data set. Okay. So moving ahead, I would like to ask to the audience, how many of you are aware about this principal component analysis? Is this a new term for you or new concept for you, or you were aware about it? So if you could answer in the chat box, whether your people are knowing about this principal component analysis, yes or no? Okay. File new concept. What about the rest? No idea, new concept. Okay, fine. Okay. So moving towards directly towards the running the code in the R software, I will give you a brief description about this principal component analysis. Why it is being used, what is the meaning of this, how we can use this particular thing, what is the basic calculation for it. Okay, so in a small descriptive way, I will try to complete this particular topic. So now without wasting much time, I will move ahead. So PCA or you can say it as a principal component analysis. So it is also known as proper orthogonal decomposition or Carnot love transform or Hotelling transform. These are other names of PCA. So this PCA actually helps you to find out the most dimensions of your projects and it make your analysis easier. Imagine you are working on a project, okay, and a lot of different variables you are having. Then what do you think? All the variables are going to give you a particular output? Obviously not, right? So at that time, not all the variables are going to give you the importance for your analysis or important output. Some of them will be a key driver of the pattern and you are interested in that particular thing. And while others might not contribute at all. So in this case, how you can reduce your variables, how you can reduce your data set, PCA comes into this play. Okay. So it helps you to shift all these variables and find out which one really matters in your data set. So you can simply think like a PCA is a tool which can simplify the complexity in a simple language. It reduces the number of variables by combining them into a smaller set of important ones. And now this new smaller set of variables which you will derive after your analysis, they are called as PCs, principal components. And these PCs retain the most important useful information in it. Okay, so here you can see this diagram, like we are having this particular data set uh, plotted over this graph. You can see all scattered and some variables. It is more than this. After doing a complete analysis by developing your PC, you are reducing the data set into a linear form over the graph. So you can see they are arranged over the graph in linear formation. So this PC is just what? Reducing the dimensionality. So with this reduced dimension, you can explore and increase your data set without wasting your valuable time. So it is that's why called as dimensionality reduction process. It reduces the data sets and many variables and compress them into a smaller set of variables for your easy interpretation of your data. Okay. So now moving to the next slide, if we talk about PC application, then where can use these particular statistical tools? The first thing is data cleaning and data pre-processing. You can monitor and visualize dimension data in 2D and 3D dimensions. This helps you to compress the information using its quality, as I already told you. Means we will reduce the large number of variables and make it into a smaller data set. But that doesn't mean that your important quality information will be lost. No, it will not be lost. Okay. Then simply but one more thing I have mentioned here is it minimizes the most significant variance of dimension. That means it can omit the external factors, the noise factors or the PT. Okay. Now next is when we can use this PC functional factor analysis. Okay. Some we are confused when we are having lots of data sets and we think of like why we will whether we can use this PCA or not. So sometimes we are completely clueless about it, right? So for that, I would like to say there are 3 points which you have to see in your data set. For example, that is firstly, if you want your data to reduce number of dimensions, like, next, if you want to categorize the variables, categorization of variables means you want to categorize the dependent and independent variable in your data set. And third thing is to when you want to eliminate the noise components in your dimension analysis. So at that time also you can use your PCA. That means any external factor you want to omit. So these are the 3 points on the basis of which you can decide whether we can go for your PCA analysis in your data set or not. Okay. Now you must be wondering whether this PC strategic analysis is really helpful. Of course it is. Like for example, tell me what if you need to handle 100 variables? Okay, let's talk a real-time example to understand this. Let's not scroll the slide and discuss a little bit. Okay, imagine you are a data scientist and you are working in a big store, and your job is to figure out what kind of things customer like to buy, so that you can make a better ad out of that. Okay, that is one example I'm trying to give. So you have a lot of information like uh, how old your customer is, how much money they can make, how often they shop, what they buy, and many more informations you are having. So by looking at this information, can you able to give a simple conclusion and make a bet out of that? Will it be possible when you are having so many information? I am giving you 4 examples, but there are like 100 options you are having about your customer. So how you are going to conclude in one line? Will it be possible? Will it be possible? Anyone from the audience can say, having a lot of information about your customer, who wants to add, and to conclude what option somebody written it. No, it's actually very tough. My voice is not clear. Now clear? Let me know. I would be more audible. Yeah. Can you guys hear me?
Yes, ma'am.
Okay. Thank you. Yeah. Let's coming back. So right. So when you are having lot of information uh, about a customer and you want to conclude something so that you can make a better ad. So at that time, such type of information at a once is really tough. So that's where this PCA comes into the picture. PCA helps by finding the most important directions called as PCs. This PCA's principal components, they are very important. So these are not just a single type of information like age or money, but they gives a combination of information. For example, you are going to get in your analysis, you are going to get different PCs like PC1, PC2, PC3. I will show you in the upcoming slides. So this PC1 actually gives a combination of information like amount spent by the customer, how often they shop. So that PC1 will indicates the overall engagement of the customer. And PC2 may mix the age of the customer, what they buy. So all that will be reflecting a different lifestyle segments. And usually when you get multiple PCs, the first few PCs actually gives you the proper information. You need not to cover up all the PCs. You have to visualize only this first or second or third PCs. They give you the total information about the data set. Okay. So by this, you can plot your graph and get your information. Now let's move to the next step, that is step by step of PCA. So this step by step is being required for basic calculation of your PCA when you are doing it manually. So manually, how you are going to do this PCA analysis? That is a basic thing. Before moving towards the analysis in the software, we should understand how to calculate it manually by taking a small example. Okay. So the first step is standardize your data set. That is to make it simple, normalize your data set. Then calculate your covariance matrix. Then calculate the eigenvalues and the eigenvectors. And then finally recast the data by calculating your principal components. So these are the steps involved in PCA calculation. I'm going to explain one by one each steps. So the first one is standardize the data set. Okay, let's say we have a data set with 4 variables and 4 observations, as you can see in this particular table. So the first step is to standardize. Standardize means transforming all the variables as they will have a mean of 0, standard deviation of 1, and hence your variance of 1. That is important. Now you must be thinking, why we need to do this standardization? Anyone is knowing why we need to do a standardization of the data set? Anyone can respond? If you don't know, then you can also answer no. If you know, you can say it in a single line why we are standardizing the data sets. Okay, fine. So the reason, okay, to minimize the error, of course. So the reason is it is done to ensure that there is no imbalance in the contribution of the variables. Means there should not be any biasness of the contribution of the variables due to their unit differences. Otherwise, the variables that have higher variance, that they will contribute more than the one with the lower variance in identifying the principal components. Although it does not reflect the reality, right? So that's the reason to do this unbiasedness and imbalance in the contribution of the variables to develop the PCs. We need to standardize our data set. And to do that, we have a basic formula, like here it's written. Z is your symbol is for standardization equals to value minus mean upon standard deviation. So first thing you have to do is you have to calculate the mean of this particular data set. Okay, like x1, x2, x3, x4, you will calculate the mean for them. Okay, after that you have to calculate your standard deviations for the data set. And finally, you have to put the values. Values means like 0.39. Here it's 0.39 minus whatever is the mean divided by standard deviation. Like this, you will calculate your uh, values. Like I have done this here, you can see the mean I have calculated for all the variables, standard deviation again we have calculated. And after that, we have finalized by putting the values in the formula and calculated the standardized values. So if you compare now, if you can see these values and compare it with these values, you can see that the variability does not vary much. The values does not vary much for the sample. Hence, standardization is not much in this particular example, but however, for the sake of illustration, the variables were standardized. Okay. So this is our first step. After that, we have to calculate the covariance matrix. So, what do you actually understand by this covariance matrix? Why we do this? We have to develop this, calculate this particular covariance matrix just to observe the association between the variables in our data set. Association means simple correlation. Correlation between the variables we want to study. Therefore, we are calculating this particular covariance matrix. So computing this covariance matrix actually helps you to identify the relationship between the variables. That means how the variable vary from the mean with respect to each other. Okay, so here you can see this particular formula of covariance. of x and y, we have summation of xi minus x mean multiplied by yi minus y mean divided by n. So n is your number of observations. So here when you are going to put your values, you are going to get a table like this. Okay. If you want to calculate the values for x1 and x1, then here it will be xi minus x mean ka whole square divided by n. But if it is two different variables like x1 and x2, then you can put here the second value. Okay. So like this, you can calculate this particular covariance matrix. Right. Now the question is, what you are going to interpret out of this table? What this particular values present in this table is saying? Can anyone say what is this covariance matrix is telling you? What kind of information they are telling you? Anyone? See, this covariance matrix is simply just like your correlation table. Okay. Here you can see the positive values. So this positive values actually implies that the variable pair is positively related. So in other words, if I say when the magnitude of one variable tend to increase or decrease, the other does so. So here, if you are having a positive value, that means increasing your one variable like x1. If I talk about 0.02, so if I increase this particular variable value, then this particular x4 will also increase. This is what the positive and positive correlation used to tell. Okay. Similarly, the negative covariance. So negative covariance implies the variable pair is inversely related. What do you mean by this inversely related? That means when the magnitude of one variable tends to increase, the other tends to decrease and vice versa. Okay. Right. Some of you have written the correct answer that this particular table actually tells you about positive and negative correlations. Okay. So now in our case, you can see that the variable 3 and 4, they are only negatively correlated, whereas other variables are actually positively correlated. Okay. You can see the negative values here only in these two variables. Right. So after calculating our covariance matrix, now we will calculate our eigenvalues and the eigenvectors. So to determine any principal components, we need the eigenvectors and eigenvalues first. And these eigenvalues and the eigenvectors inform us about the direction and the magnitude of spread of our data. Okay. So here as I have written, eigenvector is the principal component represent the direction of maximum variance in the data. An eigenvalue represents the amount of variance in each component. There is a small difference. Okay. So as I earlier discussed, that the principal components are the new variables that are formed by the linear transformation of original variables, which I have shown you in the first graph. So this eigenvectors gives the weights to be used in this linear transformation. An eigenvalue tells how much variance is explained by this newly transformed variables which are present in the principal components. So to calculate this particular eigenvalues, we are having this formula. Okay. So what this formula is saying, 0 equals to dt is determinant of covariance matrix. And S here represents your covariance matrix which you have calculated in the previous slide. Then minus delta. Delta is your eigenvalue and I is the identity matrix. Identity matrix is nothing. It is just like 1001 matrix. So if you put the values in this particular thing, then you can in this particular equation, then you can get a value of lambda. So for you, I have calculated here the value of lambdas. Lambda 1, lambda 2, lambda 3, lambda 4. Okay. After calculating your eigenvalues, you need to calculate the eigenvectors. So for that, we are having this formula. S is again your covariance matrix minus delta, that is your eigenvalue. So you can take one eigenvalue at a time. Like you can take lambda 1, lambda 2, lambda 3, one at a time. Then your identity matrix, and this U represents your eigenvectors. So by putting this particular value, you can get a table like this for your eigenvectors. But the work is not yet done. What you have to do is to calculate the PCs, we need to consider the largest eigenvalue. Like here, you can see your largest one is 1.66 compared to 1.01. You can take these two values to calculate your PCs, the largest eigenvalues. Okay, lambda 1 and lambda 2. And after calculating your eigenvectors, you have to calculate your unit eigenvectors, as we are more interested in calculating this unit eigenvectors. And after calculating your unit eigenvector, finally you can calculate your principal components. So this is the simple steps we use to follow for the calculation. And here also in this table, as I told you earlier, that the eigenvalues are ranked based on their highest to lowest, and we have taken only the two which are showing the highest value. That means the lambda 1 and the lambda 2. Similarly, here also, based on our result, we will choose two eigenvectors. That means E1 and the E2. Okay, which you have taken here, right? So from here, we will calculate our PCs, and that will be the final step. So these are the basic calculation steps we are using for our calculation of PCA, the basic steps. Okay, these are the 4 steps. Is it clear, or anything I need to repeat? Yes or no? Yes, please, somebody respond. Okay, repeat. Okay, fine. See, the thing is, in PCA, you don't have to go for all the basic manual calculation, because the manual calculation is quite tough, and these are the 4 stages where you have to do the calculation. Like standardize your data set, simple. That is, this is your data set, and you want to standardize your data by using this particular formula. After calculating that, you have to calculate your covariance matrix. This is like basic mathematics. So if you put this covariance matrix formula, you will get a table like this. After getting this covariance matrix, you will develop the eigenvalues and the eigenvectors. So for calculating the eigenvalue, you have to use this particular formula. Okay. From here, you will get such type of values: lambda 1, lambda 2, lambda 3, lambda 4. But being lambda 1 and lambda 2 is showing the maximum value, we will consider these two eigenvalues for our further PC development. Our major thing is you have to develop at last the PC principal components. So this is the whole process we are doing. Okay. So after eigenvalue calculation, calculate your eigenvectors. So you will get a table like this after calculating all your eigenvectors. And here also we will take only E1 and E2 because they are showing the maximum variance, because they have been developed from lambda 1 and lambda 2. And lambda 3 and lambda 4 will give this E3 and E4. And they are giving lesser value, lesser variance. So we are excluding this E3 and E4. We are considering only E1 and E2 for our PC calculation. So from here, we can calculate our principal components. These are the steps involved in the PCA calculations. Okay. So far, actually, we explained the PCA theoretically, but we have not shown you the use of this PCA in practice yet. So let's see one application, a real-time example I'm going to take. It is actually a data set of pizza data set. I know by looking at the word pizza, it would be looking a little bit interesting. I think it will give you a layman understanding. That's why this example is being taken. So the data set actually contains the ID number, the first column, then the brands, then the nutritional content for actually 300 samples and 10 brands. Though I haven't mentioned here all the data, it will be a huge thing if I will mention all the brands. Okay. So we are having a 300 sample and 10 brands, and these are our traits. That is, uh, you can see this. This first column is your moisture, then this is protein, fat, ash, sodium, carbohydrate, and calcium. Okay, and these are your brands. So we have this particular data set. So based on this given data set, can you tell which brand is best for you? By looking at 10 brand data, though I haven't mentioned the whole values, but you can imagine I have written up to 6 brands. So the values are this, this, this. So among the 6, can you able to tell which brand will be the best brand and can say like such type of things? Of course, by looking at this particular data set, you cannot say anything. Okay, just a numbers and values, and it's very difficult to say by this naked eyes which one is which brand pizza is good, right? So for that, we are using this actually principal component analysis. So to calculate the principal component analysis to principal components, we need to understand the meaningful association among the nutrients, and the variability of the brands can be explained by few pieces of informations. Like firstly, let's take the correlation matrix. So from here we can give a little bit of conclusion, right? So in the correlation matrix, you can able to understand which trade is correlated to another trade in which way, whether it is positively correlated or negatively correlated. So here in this table, if you can see that the moisture, you can see this first column, first row, moisture. Hmm. So moisture here it is actually negatively correlated with carbohydrate and calories, right? Then protein, you can see it is positively correlated with fat, ash, and sodium, but it is negatively correlated with carbohydrate. Then fat, similarly, you can see it is positively correlated with ash and sodium and negatively correlated with carbohydrate, right? So in this way, you can interpret your correlation matrix of this particular data set. Now moving ahead, let's see this table of proportion of variance. Actually, such type of tables are being derived after analysis in your software. If you're using a software, the software will give you such type of tables. I'm just giving you a small tables. I'm showing you here a small table. But if you are having a huge data set, you are going to get a plenty of tables. Means number of values will increase. Okay. So here, what actually this table is interpreting? What actually it's saying? What is the meaning of this proportion of variance table? So here you can see the first two PCs, the PC1 and the PC2. Total accounts for 92.3% of variance in the data. How I am saying 92.3%? Simply you add these two values and you are going to get it. Okay. So here 92% is a considerable amount, and you can say it is like sufficient to retain the first two PCs in the analysis. Okay. So here we can say that first 2 or 3 PCs gives you the maximum amount of informations. So this proportion table is just to tell you about the amount of variance present in that particular data set. Now it's the time to see the relationship between the retained components and original variables. So this plots showing you the relationship uh, called as loading plots, or you can also say the weight plots. And it is used in the linear transformation of original variables to the PC. So based on these loading tables, what you can interpret is PC1 represents the rich in fat. You can see the values. The PC1 actually showing the fat, ash, and sodium values are quite high, right? And poor in the carbohydrate content, because here you can see it is negative value. Whereas PC2, you can see this PC2, it represents the rich in the calories. You can see the value of calories, and poor in moisture and protein, right? So these are the factor loading tables which you are going to get, and here you can interpret all these things. Though I have taken here 2 PCs, but in your data set you may get more PCs also. So accordingly you can do a little bit of interpretation. Okay. Next is this is a variable plot. This also you are going to get in your analysis. So now the question is how to interpret this particular graph. Anyone is having any idea how to interpret this particular graph? Yes, please respond. Okay, no, right. So here you can see what all variables we have taken. Variables means the traits or the features, whatever we have taken for our study, we have plotted it here in this graph. So in this you can see here we have taken the 2 PCs, PC1 and PC2, and done this plotting. Why we have taken only these 2 PCs? Because they are contributing the maximum variance like 59.6 and 32.7. So all total it is contributing maximum variance to the data. So that's why we are taking these 2 PCs and done the plotting of all the variables in this particular graph. Okay. So for interpretation, the vector projections are made use of. Means vectors means these arrows. Okay. So suppose this is your line, okay? This particular line is considering your PC1. So all the variables falling here, you can see. So PC1 is actually concerning the projections of calcium, fat, sodium, ash, protein, and moisture. Okay, because it is falling towards this side of this middle line. All these variables are falling here, right? And if you see this particular carbohydrate, it is falling in the opposite direction to this all these traits. So what we can say that PC1 actually comprises of all these variables, but it is low in the, it's showing less variance in the carbohydrate. Similarly, if you look at PC2, these are the dotted line of PC2. So all the variables which are falling above it are considered under PC2. For example, your calcium, fat, sodium, all these are above the equator line. You can say the PC2 line. So PC2 is contributing these variables. But ash, protein, and moisture, these 3 variables are not showing greater variance or greater contribution in PC2. They are low because they are falling in the opposite direction. So this is how you can interpret your variable plot by looking at the vector directions and their magnitude. Okay, so from here, if relatively, if the relativity increase magnitude are taken into account, then we can say that the PC1 refer in the richness in the fat, sodium, ash, and protein, and lack in carbohydrate. But PC2 rich in calories and lack in the moisture because this PC moisture is falling in the opposite to the middle line. Okay, okay. Now it's time to use this particular inference in deciding which brand actually work best for you. Which brand is best for you? How you can decide that? So by looking at this particular biplot, we can conclude it in very better way. So biplot is what? Biplot is actually a scatter plot. You can see all the things are being scattered here. Brands, then variables, everything are being scattered. So it is actually a scatter plot. So in this special plot, the original data is represented by principal components and they explain the majority of the data variance using this particular PC scores. So uh, so here by looking at this figure, how you can interpret? So here you can see the axis shows the PC scores. This is the axis. This is y-axis. This is x-axis, and they are showing the PC scores. Okay, and the point referred to the individual pizza samples are colored by the brands. Here you can see these are the brands. This is A brand. Then this blue one is some G brand. So like this, these colorful things are the brands. So from here we can say that if you want a fatty, crispy pizza, then the brand A is a good option. Why I'm saying this? You can see this. This is brand A, and this particular vector of fat is facing towards brand A. So if you want a fatty, crispy pizza, then you can take this particular brand A because the vector of fat is facing towards the brand A. Okay. Similarly, if you want a soft and non-fat pizza, then you can use the brand I. Here you can see this is brand I. It is a non-fat pizza. Why I'm saying this? Because it is opposite to the direction of fat vector. Fat vector is this side. So if it is this side, this particular brand will be fatty. And opposite to it, this particular two brands are there. They will be non-fatty. And if you want to prefer a balance of nutrients, then brand B and J. Again here, B, this particular thing, and J, you can say this one. Here you can see all the variables are falling here. Maximum variables, nutritional variables are falling in this particular B and J things. So that's why I'm saying if you want a nutritional pizza, you can go for this two brands, B and J. Okay, like this you can interpret. If you want more carbohydrate types of pizza, then you can use this blue and this again sky blue H and G. H and G types of pizza brand you can go for. So by looking at all these things, these variables and the brands, by doing this by plot, it is very easy to give a particular interpretation. Okay. So in this way, uh, we use to interpret our PCA biplots. Okay. Is it clear? Yes or no? Thank you. Okay. Right. So moving ahead, again, we are having this application of PCA, which I have already discussed. A little bit of new things I'm telling. Like these days, we are going for image compression. Actually, PCA reduces the image dimensionality uh, by without losing the valuable data. So it actually helps create a compact representation of image. and it makes them easier to store and transmit. Next is your data visualization also. So like any 2D and 3D visualization plots you can develop by using PCA. Next is you can omit the noise also. You can remove any kind of external factors from your data set. And last one is your feature extraction. So feature extraction is like it means that you can produce new variables that are linear combination of original variables. So this means PCA can identify the most important variable in your data. So like this, these are some applications I talked about. Okay. So now what we have covered till now is simply what is PCA, why is it being used, when to use it, and how to interpret the graphs and the tables in your PCA, and the basic calculation manually how you can calculate your PCA. So till now I have covered this much, and now I am moving ahead how to calculate this in your R software. Okay, uh, my screen is visible. Yes, can you see the data set? Yes, okay. So here I am having a data set. Okay, so you can see the first one is your genotype. It is actually a plant breeding data set. So I'm having a genotype. Then replication, I'm having replication 1, 2, 3. Okay. And genotype, similarly, for each genotype, I'm having 3 replications. Like second genotype, again, I'm having 3 replications. And these are my variables. These are my variables, or I can say the traits, plant traits. Okay. So how much data I am having? You can see it is actually 30. 30 genotypes I'm having. 30 genotypes, each are having 3 replication, and these number of variables I'm having. So to run this particular data set in the R, what I will do is, see, I have written the codes already. I will also share all these codes with you. I've written it already because to reduce the time. Okay. So here you can see this R workplace. So first thing you have to do is you have to import your data set. So just don't see all these all these codes right now. It will be difficult for you to understand. So let me explain you one by one, and then you just see all these things. The first and foremost thing is you have to import your data set. For importing your data set, either you use the code or you do it by manually. So how many of you are knowing how to import the data set? I hope in the previous lectures you were aware about it, how to import your data set. Yes or no? Okay, great. Happy to know that you guys are knowing, right? So either you use this particular thing, you can see here, it's written import data set, right? If you click this, you will see from Excel. If you open this, you can browse your data set here, and from whatever drive your data set is there, browse it, click it, and you will see your data set in this particular space. And then finally import. Okay. One way is this. Another way is to use the code. To use the code like this. Forget about this one. See it from 18th line. Set WD, then your folder name, like drive name, sorry. Like my drive is F, in which I have kept my data. And in my drive, I have made a folder of SR, so I mentioned that. Okay, similarly, you can also write your drive name, like whatever, D, E, F, whatever, and then your folder name. In shortcut, I'm keeping so that there should not be any kind of error while running the codes. Okay, then run your library read Excel. This library will read your Excel file. Okay, after that, I have given a name like a data to my data set. Okay, equals to read Excel, and this is my data file, which is BD. Okay, dot Excel. It's a Excel sheet. Then in my Excel sheet, if I have shown you here, you can see in my Excel sheet the file name is BD, and here I am using the sheet 1. So to read it, I have mentioned here sheet 1. Okay, so this much code is going to import your data. Just you have to change your drive name, then your folder, then what is your Excel file name, then in which sheet you have kept your data which you want to run. Because in Excel file we are sometimes keeping multiple sheets. So which sheet you want to run, that you have to put here. This much is clear how to import your data by using the code. Okay, great. So after importing my data, I can also see that glimpse of A data. Okay, so for that I am writing this glimpse bracket. I have given the new name A data to my BD data. BD is my original data, but I have given it as a name as A data. So you can write glimpse A data, and you can able to see your data set. Okay, so this is how you have to import your data. Okay, just select it and run. Right. So now you can see here my data set is being visualized here. Okay. So after this, you have to run some libraries. Okay. So for that, you can go up. I have mentioned a few libraries which are being required for your PCA analysis. Like first one is to read your Excel file only, and the remaining tidyverse, then facto extra, facto minor, metan, and R color brew. R color brew is for color palette. Just like whenever you go for any construction activity in your home and you want to color your out hall, then this Asian Paints used to give you a color palette. So similarly, there is one color palette is there in your R software to color your graphs. So whichever color as per your requirement, you can select by using this particular library, R color blue. Okay. So here, firstly, you need to run all these library, and before that, you have to install all these libraries. So for that, I hope you guys are knowing how to install the packages. Yes or no? Yes, anyone can tell me if you know how to install the package? No. Okay. So here you can see the packages, right? It's written here. So these are actually the libraries, and here you can see install. If you click on install, and here it's written packages. If you write this facto extra, facto minor here, then click on install. In this way, you can install your library. All these things are installed in my R already, so I need not to do it again. Once it is installed completely. So you can do it in your R. Like you want facto extra library, right here facto extra, and then click on install. Simple. That's it. In this way, you will install all these libraries in your R. So once it is being installed, then write this particular function, library bracket read Excel. Okay, and then run it. Write all these library all together and select it, then run. Okay. So in this way, all my libraries will be run, right? Now, after that, what you have to do is, I want to make my genotype and replication into factor. I have to give a command like my genotype and replication column, that is my first two columns, need to be converted into factors. So for that, I have written here my data set name as A data. And similarly, I have mentioned all these codes. I'm making the genotype as factor and replication as factor. Okay. So again, select this. You can also glimpse this particular A data after making it factor. So run. So now you can see here genotype and replication FCT it is written. Now these two columns are converted into factors. Okay. After that, what you have to do is now come to this particular code. Uh, so here what we are doing is we are making our whole data set as numeric. Okay, so for that, what I am doing is we need to convert our first column, which is genotype column, to the making it as a row name. Okay, so for that, again, I am putting my A data name of the file, and the remaining things will be just the same. You need not to change. See, this is also for your data set also. You don't have to change. You have to simply select and run for your data set also. But the only thing is you have to arrange your data set in this particular format. Otherwise, what will happen? Suppose I have written here as genotype, but you will write it here as gen or XYZ, whatever. And here it's written as rep, not whole replication, but you will write replication and you will use this particular code. So error will come. Why the error will come? Because here I have written genotype. In my Excel sheet also I have written genotype. When I have written rep, in the same way in the code also I am writing rep. Okay. So these things you have to change in your codes according to your data set. But the remaining things will remain the same and you can simply run it to get your data analyzed. Okay. So here now I am going to make my data set numeric by selecting all these things. Run it. Okay. Now you can see it is all numerical data set has been defined. Okay. After that, what I'm trying to do is, here you can see this replication column. I want to remove this replication column and making my replicated data into a mean data. Here, what data I am using is a replicated data, right? So I want the mean of this, like R1, R2, R3 divided by 3. I want a mean data. So for that, I am using this particular code. So here again, I am putting my file name and minus replication. Minus replication means I'm removing my replicated data. Okay. So simply again run it. So after running this, you can see my replication here at this place. You can see this. After running this, you can see this particular thing. All these are now replication has been removed, right? Earlier replication was there. Here replication was there. Now it has been removed and only mean data is being observed. And PCA is foremost thing. PCA is usually being calculated from the mean data, not from the replicated data. So now, after making our data set properly standardizing it, we have to run the PCA. So this is the simple code which I have taken directly from the Google. It is all available. All these codes are actually available in Google. Okay. The only thing is we need to change a little bit of things like here I have changed only my file name like A data and the remaining thing I have copied from the Google. So you can also do the same. So run this PCA. Okay. Then calculate your eigenvalues. Run it. So here you can see this particular thing. Dimension 1, dimension 2, dimension 3. These are your principal components. PC1, PC2, PC3, PC4 and so on up to PC11. And these columns you can see these are your eigenvalues. Next is your variance percentage. 32.21 variance is observed for PC1. 20.63% is being observed for PC2 and so on. These are your variance percentage. And this is your cumulative percentage. Means PC1 is giving you 30. But altogether PC1 and PC2 together gives you 50 percentage, 50.85% of variance, right? So here, if your eigenvalues which are greater than 1, usually we take those values into consideration. So up to PC4, we are going to consider the PCs for our study. We will not take all PC11. The PC11 is giving us very less information. Up to PC4, because the eigenvalues are greater than 1, up to PC4, we will consider the values. We will consider it for our study. Okay. So this is your eigenvalue tables, which I already told how to calculate. Okay. Now moving ahead, here also you don't have to change anything. You have to simply run, and this gives you a scree plot. Okay. Yeah. You can see the scree plot. So this scree plot is telling you what? If you will recheck with the previous table here. Here you can see PC1 is giving 30.21 value. It's written here 30.21. Next is PC2 is giving a percentage variance percentage of 20.6. The same thing is written. So this tabular value is being represented by using this scree plot. So in this you can see this particular line. So up to PC4, your line is almost straight. After that, it is like declining. You can see that. So this declining is nothing. It is saying that the PCs, later PCs are contributing less in your data set. Only the first 4 or 5 PCs are giving you the majority of your answers, majority of the percentage of variance. Okay, so scree plot is meant for this. Now you must be thinking how we can save this particular plot, right? So for that, you have to do is, here you can see export, click on this. Here you can see this save as an image, save as a PDF. So if I want to save it as an image, so here in my F directory, that means F drive in my own folder in which I am having this particular data set, it will be going to save there automatically. Okay, then you can click here as save. You can change the width and dimensions also. If it is very clumsy, like here the data is not that much, so it is quite good to see. But if your data is too much clumsy, you can change the width and height. Okay, then you can save. Right now I'm not saving that, but this is only for your information. Okay. So this is our first plot of PCA, that is called as scree plot. It tells you about the variance percentage. And obviously, which principal component is giving you maximum information. Next comes your individual plots. So here again, you don't have to do anything. Simply, you have to write this particular code. No need to change anything in this. Okay, and run it. So here you are going to get the factor loadings. Now you can see this particular workplace here, dimension 1, dimension 2, dimension 3, 4, 5. So these are the 5 PCs. Okay. They are giving you the factor loadings for these 5 PCs, because here you can see up to 5 PC you are getting a value of 7.3, right? 1, 2, 3, 4, 5. Up to this, the scree plot is straight, and after that there is a elbow bending. So that's why the loading graph is also showing up to 5 PCs. But you can exclude the 5th PC because your eigenvalue should be greater than 1. And PC5 is having a eigenvalue of 0 or something, 0 point something. Okay. So this is your factor loading. So for a DFF, you can see which PC is contributing more by looking at their value. Then for the second trade, PVB, which PC value is showing the maximum trade, maximum contribution. Like this, you can interpret this particular table. Okay. This is factor loading table. Now comes your visualization of individuals and variable graphs. Okay. So here you can see all these are completely taken from your internet only. But here one thing I would like to say, here can you see this blue pal? This blue pal is actually telling you about the color palettes. Okay, for color palettes run, what you have to do is just have to write this code. Wait, I will write here like display. Yeah, it's here. Display view all and close bracket and run it. You can see this. Can you able to see this color palette? Yes or no? You can zoom it and see. This color palette is visible. Yes or no? Okay. So here you can see if you want this particular color, like the first one. So here there is a code written here, YI0, sorry, ORRD. Okay. So by using this particular code, and if you will run your graphs, you can get this particular color, the first one. Similarly, whatever color you want, the codes are written here. You can choose according to your choice. So whatever I have chosen is PRGN. Okay. And in PRGN also, I have taken 8, 9, 10. Means in PRGN, let's move to PRGN. If you see this particular band, there are so many colors in that. Okay. Which color you want? So you can say 1, 2, 3 I want. Okay. So you can take 1, 2, 3. Similarly, whatever one you want, you can count from starting to the end, and whichever number you want, you can directly write here. Here, 8, 9, 10. Like I have written PRGN 8, 9, 10. Similarly, you can write for your. So simply you have to do is you choose your color and the remaining things will remain the same and run, select it and run. Will come. Yes. So here, this is your variable graph. Okay. Between two dimensions means PC1 and PC2. PC1 is contributing 30.2 percentage and PC2 contributing this. Okay. You can zoom it and do a better interpretation also. See, these are the PC1 and PC2 and the same variable table graph as I showed you in your here, PPT. So from here you can do your interpretation of your data set. Okay. Next graph is your bike graph. So this is your bike graph. Just simply here also you can change your colors and codes and the remaining thing will remain the same. This is your biplot graph. Let's zoom it. It is biplot because you can see the variables. The variables are in alphabets and these dots like 17, 22, 20 and all these things. These are my genotypes. So I am plotting both my genotypes and variables in one graph for my better interpretation, like which genotype is good and which genotype is good for which trade. So from here I can do my interpretation in a better way. Okay. So this is the biplot. And the last one is your core plot. This is a supplementary thing. If you want to, it's okay. If you don't want, it is also okay. Because up to this biplot, we are getting maximum of our information. We can do the better interpretation. But this correlation plot is also there. For that, what you can do is you run the library like core plot, just your simple correlation in PCA. So select it and run. Okay. So here you can see this particular core plot. Core plot actually says what? See, here all the traits are written here. That means all the variables are written here. These dimensions are your PC, and this is your correlation scale. Okay. So suppose I'm taking the PC1. So in PC1, you can see which one is showing the darker bubble. Can anyone tell? Which one is showing the more darker pink ball and bigger ball? Which particular variable in PC1? Right. So here you can see this FD and AFW. These two balls, if you will see, they are showing the darker as well as bigger balls. So what I can say from here is PC1 is showing maximum correlation in this FD and AFW trade. Similarly, if I go to PC2, I can see the bigger ball in this particular FYP. So this is showing the greater correlation in PC2. And then in PC3, you can see DF is showing. Then in dimension 4, you can see PC4, you can see TW is showing the greater correlation. So like this, you can interpret this particular core plot. Okay. So till now, we have discussed about your PC and the codes. Anyhow, if you need the code, we can also share. And if you have any of your own data set and you are facing any problem, you can also share that. We will do it for you. Okay. So is it clear till now, whatever we have covered in PCA? Yes? Okay, fine. Anyhow, if you have any problem, you face any problem, you can directly post in the group, or you can mail us also. Okay. And next, I would like to cover up a little bit about the cluster analysis. So in cluster analysis, actually, why we do this? Usually, we do this cluster analysis for our plant breeding experiments to assess the genetic diversity. Like in a layman language, I know many of you are from different background, you're not able to understand that particular thing. So in a layman's thing, I will say, if you want to develop a hybrid, okay, so to develop a good hybrid, you need good parents. And to get a good parents, you need a diverse genotype, diverse parents to cross with. Okay, so if you get do a crossing of a good parents, then only you are going to get a good hybrid. So for that, what we do is this clustering thing we are going to do. Okay. So actually, this genetic diversity helps. It is play an important role in the plant breeding because hybrid between the lines of diverse origin, they display the greater heterosis while compared to the closely related parents, right? So for that, we are doing this particular cluster analysis. Again, I'm using the same data set. Okay, I'm having a data set, this one, and among this data set, among the all 30 genotype, which are closely related, which are distantly related. We can able to know this only by the cluster analysis. So for that, what we have to do is, firstly, we have to develop a matrix that is called as Mahabelanobis D square matrix. There we can get the D square values. Greater the D square value, greater the distance between the genotypes. So for that, I am running this, I'm giving the new name to my A data as a new data. Okay, I'm not putting the same name. Like earlier it was A data, now I am giving it a new name as a new A data. Okay, now run this matrix. Simply I just change the name and running this matrix. It is actually a huge matrix. So here you are not going to get the full table. You can save it in the Excel. So here you can see this. This is a huge table coming, not fitting in my workplace. This is this type of table I will get. This is actually a D square matrix, D square values, distance matrix. You can say that. Okay. After that, I have to do the clustering. So I'm using this particular code for my clustering, that is H cluster and distance matrix. All these things I'm writing, and I want a clustering of by using the word method. Okay, there are different methods are there, so I'm using the word method, and I want a cluster of 7. If you want to edit, you want a cluster of 4, you can change the value from here like 7 to 4. Like this, you can change depending upon your need. Okay, so here by running this particular code, I'm going to get my clusters and dendrogram. If you heard about dendrogram ever before, yes or no? Dendrogram, have you seen? Yes, okay, great. Okay, so similarly here in this, by running this particular code, I'm going to get my clusters as well as my dendrogram. The only thing I'm doing here is I'm putting my column name as genotype, and remaining things will remain same for all of you also. You can change the clusters, you can change the method, which method you want, and remaining things you need not to change. Okay, run this particular code. Okay, so here you can see the clusters. Can you see this? Genotype 1, yeah. So here you can see the cluster 1. Actually, it's not genotype in 1. It is actually cluster 1. Cluster 1 consists of genotype 1, 2, 3, and 17. Then cluster 2 consists of 4, 12, 18. Cluster 3 consists of 5, 7, 8, 9, and so on. Then cluster 4 consists of 16 and so on. So I have made 7 clusters, and in these 7 clusters I have distributed all my 30 genotypes. Now what actually this cluster is saying? It's saying that like 1, 2 and 3. These 3 genotypes are like 1, 2, 3 and 17. All these genotypes, if they are falling in one cluster, that means they are closely related. But if I compare genotype 1 with genotype 16, 1 is falling in genotype 1, cluster 1, and 16 is falling in cluster 5. So what I can say is these two genotypes are distantly kind of different from each other. They are diverse. They are not having any close genotypes. They are different from each other. They are diverse from each other. So if I take these two genotype and go for crossing, like one cross with 16, I may get a good hybrid. Okay. But from here, clustering, we cannot directly say this thing that if I take 1 and 16 into the hybridization program, it will give a good result. No, I cannot conclude this particular line here. But here, from here, cluster analysis, I can conclude one thing, that the genotypes falling in the same clusters, they are closely related compared to genotypes falling in the different clusters. So this much I can say from here, from this table. Okay. Now for dendrogram, you have to run this particular library. For this, firstly you install it and then run this dendrex library. After running this, just run it. Okay. After running this, you can simply run the remaining codes. Up to this much, you run this. Okay. Yeah. Here you can see this particular dendrogram. Right. So here you can see I have made a dot type of things also. so that it will be more clear how many clusters have been developed. So it is 1, 2, 3, 4, 5, 6, and 7. So all the different colors, if it is visible to you, you can see they all 7 clusters are arranged in a different colors. You can also highlight them by using this particular code. Like if you will run this code, it will be more distinctly colored. Yeah, you can see this, right? Now it will be more distinctly visible how many clusters are there. 1, 2, 3, 4, 5, 6, and 7. 7 different colors and 7 different clusters. And all our genotypes are lying here. You can see all our genotype numbers are mentioned here. So like this, you can also prepare your dendrogram by using this particular code. And simply you don't have to do anything. Except if you are putting your cluster number, you are changing in the upper codes, then here also you have to change. Otherwise, there will be a mismatch. Suppose in the upper side, I am putting 4 as my cluster. Here also, I will put 4. Okay. So this is what your cluster analysis used to say. And after that, if you want to calculate your inter and intra-cluster distance, then you can further go for your another library, that is CLV library. Okay. From here, you can simply run your code and take the intra-cluster distance also. But sometimes, like some people don't require it. Simply on the it is actually required for plant breeding data set. But maximum of you would be requiring only dendrogram. So I'm not running all these codes. Simply you focus up to this much. That is up to your dendrogram thing. Okay. That means up to this much code you focus. Okay. Is it clear to everyone? Is this dendrogram thing is clear to everyone? Okay, fine. So I think, yeah, right. So if you face any problem in running the codes also, you can directly message me or mail me. We would definitely help you in your data set. Okay. So thank you so much. I think this class would be informative and something new many of you have learned. So now I'm moving, giving it to Dr. Shauna. So I think I have completed my lecture.
Okay, thank you, Dr. Rout, for taking this session. I think our participants get a very informative knowledge from the lecture, and there I think some of the participants wants some codes. So if you can provide to this WhatsApp group, that will be
Yeah, yeah,
beneficial.
sure, sure. Okay, I will share it so
Oh,
people can use it. No issue.
okay, okay, okay then. Okay, there are some question. Kindly
What
address
are the
them.
methods of visualization apart from dendrogram? See, for clustering, we have to prepare the dendrogram. There is no other option. The only thing is you can make it in a this rectangular shape, or you can go for your circular dendrogram is also available. For that, you can also take the codes from the Google. There are different types, means different ways to represent, but they all are dendrograms. Either you do it in a circular form or in a rectangular form. Okay, for cluster analysis, we have to do this. Anything else?
Okay, thank you. I think you have cleared the confusion. So now we are closing the session, and thank you once again, Dr. Raut, for taking this informative session. Uh, Thank
Thank
you
you.
all.
Thank you.